Yeah. So what I wanted to do is spend a little bit of time expanding on some of the issues which were brought up during the talks, and that is issues around timing. You know, that we talked about turning back the clock and restoring tissue, but at least in the eye, once you've degenerated the retina to a certain point and you've wiped out those retinal-tectal projections, can you really restore vision? And similarly in the central nervous system, we're taught that once you wipe out a region of the brain, it's very difficult to regenerate that. So it's like your mother told you, right? An ounce of prevention is worth 20 pounds of cure. Do we need to intervene earlier in these diseases? The other thing is location, 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 right? Like the real estate brokers always tell you. Where do you put these cells and what parts are amenable to treatment with this sort of therapy, at least in the eye and the central nervous system? And finally, understanding what we're observing. That is, what we see isn't necessarily what we get. Just because you put an RPE cell into a patient with Stargardt's or macular degeneration doesn't mean you can have a normal, healthy layer of RPE cells. So I don't know if you wanted to comment or if we want to have questions from the audience, but we're open for any sort of format people would like to do. Because I'd love to hear people's opinions. People working, especially with iPS-derived whatever, do you really think you're getting, cell replacement therapy? Or do you think there's this paracrine function which is really dominating the response to what you're seeing? You guys want to say something quickly, or do you want to?
Yeah, I would say in general, I feel like everybody knows one of the big challenges in this field, whether you're trying to stimulate endogenous cells to repair something or generate something in a dish for transplant, is how do you decide that you've got the right thing or that it's going to behave that you want it to, the way that you want it to. So absolutely. I mean, we do a lot of iPS-derived, say, organoid work in our lab. And I feel like it's just a constant ongoing struggle to decide, especially for something like, say, modeling Alzheimer's disease in vitro. At what point do you decide that you have made your cells old enough when you're trying to model something that happens in an 80-year-old human? Is it after 6 months in vitro, or do you need to go 2 years in vitro? Or that, I feel like that is one of the major challenges, is identity and knowing when to stop, or even what question to ask to begin with to profile what it is you're working with.
Yeah, I think that to me, even before going to the replacement, right? So we can use iPSC to study what's going on. So you have the possibility to have iPSC that are patient-specific. So you can differentiate them in different cell type, right? So if you have diseases that impact different cell types, you can make from one patient iPSC many different cell types and just see what's going on at different levels. So you can culture them together and see what's going on when you put those cells together. So just to understand what the niche is doing, right? So, and the other things that I think is very interesting is that with iPSC and in vitro study, what you can do is just take one type of cells that comes from a patient that has macular degeneration, and you can take other cells from a donor, like a normal donor, right? So you can take RPE from your patient and then the vasculature from a normal patient and see how you can combine them, right? So what is the impact of a normal vasculature on an impact tissue, right? So this, I think to me at least, the iPSC field is more interesting to understand what's going on than the replacement per se right now, because I feel the replacement, as in the clinical trial, right? So you replace these cells after 1 year, the cells are still there, so the patient didn't get worse, and this is something good, but it didn't get better. And also because of the somatic mutation, so that clinical trial now it's completely different one, because now the clinical trial is on using allogenic iPSC, so iPSC from other donors. So I think that there are many ways, I think, to fix the problem, but I think that understanding first what's going on between the tissues is the very good question, before transplant.
And also considering, again, largely for safety reasons, but we often start out with the worst patients, right?
Yeah.
And to expect a good result in an environment which is totally devastated, it's like trying to, sort of repopulate an area which has been totally destroyed. And that's often the case in the back of the eye from macular degeneration. There's huge deposits of the angiogenic lipids. There's destruction of the normal anatomy and architecture. So how do you get around that? I guess we'd always thought in terms of early onset, you diagnose somebody very early on, you take some of their bone marrow or blood, you make these progenitor cells from them, and then when it gets to the point where it's beginning to manifest disease, you go in, and that's when you treat these patients, not when things have gone to hell in a handbasket, so to speak. We should let the audience, hopefully people have some comments, things they want to say. Yes, so the hand went up first in the back there. Yeah.
So I was wondering, after you have the retinal ganglion cells or the photoreceptor cells made and you put them in the patient, how do you ensure that they synapse correctly? Because obviously that's completely critical to the vision being properly restored.
Yeah, well, so that's precisely my point. I guess I was probably speaking too slowly, didn't come across that once you've unwired the retina, how do you reestablish those retinotectile projections? And then you've got these second and third order neurons in the retina itself. So again, I think early interventions can be critical, but it's going to be very difficult to do those first trials where you take somebody who's barely affected, right, and start putting in trophic factors to help maintain that neuronal circuitry. That's absolutely critical to vision. Next question. Well, again, it's like the AVT trials. The patients you can ask. It depends how they're evaluated. I have patients from macular degeneration who barely could read the big E one week, come the next week, and suddenly they're reading like 3 or 4 lines better. So it's it's difficult to get objective, readout. I know Messiah. I'm not questioning the integrity of what she reported, but I think patients can be, led in certain directions. And one patient I wouldn't say anything about because that could have just happened whether they treated her or not treated her.
Yeah, no, and I agree. I mean, you need placebo-controlled studies.
Yes. Yeah, and early on it's going to be difficult.
But it's promising.
Pardon me?
It's promising.
No, it's exciting. Yeah, it's very exciting.
Yeah.
I don't mean to minimize what she's done, but it's,
Let's wait for other patients.
I don't know, did you have any experience? You alluded to how, my other question was, you alluded to how to give the cells and where to give the cells as being critical. Do you have any experience in trying different methods that were better or not?
Well, in animals we have, and a lot of people have been doing this in general. So in general, the debate is whether you want to do subretinal injection, put them where they have to go, or if it's really a paracrine effect, you could probably get away by just injecting them into the vitreous, which is a lot easier to do. And from my way of thinking, the last thing you want to do in a damaged retina is to induce a retinal detachment, which is what you do when you squirt things under the retina. Normal eyes can sort of fix that, but a diseased eye would have trouble doing that. And I know, Henry Klassen's study being doing on J-side is an intravitreal injection of neuroprogenitor cells. Well, they're not differentiating anything. They're elaborating factors which probably have an effect which can percolate across into the neurosensory retina. So I suspect would be a better way.
I have a question about, again, the immune system. The lineage negative CD44 positive cells obviously do not express HLA. You're talking about extracting autologous cells. Do they need to be autologous? Do they eventually express HLA antigens, or can you use a common source?
Yeah. So this is, the eye is a relatively immune privileged site. It's not absolutely immune privileged. And, you develop you develop disease in your eye that's immune mediated. You could probably get away with a certain amount. I know Henry's group is not using immunosuppression, I think. So again, I think it's going to be different for different cell types. That's it. Any final comments by the panel participants? Yeah.
Looking at your...
So I think the eyes do have it. I think we are ahead in many ways in terms of what we can do for clinical trials, and a number of very exciting things are ongoing now, and we'll know in the next few years what we can get away with and what we can't get away with. But thank you very much, and we'll
Thank you.